Good day, everyone. We are the Group 9. I'm Chloe Rafael, together with my group mates Nicole Riguer, Emanuel Sanidad, and Don Valerio. We're going to share our thought experiment, the iron coconut tree, or what we branded as the mang coconut tree. But first of all, we have to know what the mang in mang coconut means. So this mang is the source organism where the desired trait of this mang coconut tree came from. And to know more about this source organism, let's go to our next slide. So here's our source organism. It is the Santos Temon verdugonianus, commonly known as the mangkono or Philippine ironwood. If you guess that it means mango, sorry, but it is actually mangkono. And if you're wondering why it is called ironwood, then let's go to the next slide to learn more about its characteristics. So, Santos Dumont, Verdugonianus, Mangkono, or Philippine ironwood is considered as the hardest tree in the family of Myrtaceae, and it is the strongest tree here in the Philippines. Therefore, it is branded as the Philippine ironwood. It has a relative density of 1,452 at oven dry temperature. Why oven dry temperature? This is because this temperature makes the trees denser. It is 50 meters in height, 115 centimeters in bole diameter, and 70 centimeters thick. As a fun fact, if someone cut 70 centimeters of this tree using a saw, it would actually take at least 2 to 4 days. That's quite long, right? So, next, this tree is endemic, meaning it is only found here in the Philippines. But to be more specific, it is found in the Mangkono Triangle that composes of the Dinagat Island in Surigao, Humonhon Island in Samar, and a very small area in Babatnon. But it is also sometimes found in Palawan and Agusan del Sur. Unfortunately, any part of this tree is inedible and it is not suitable for medicinal use. But because of its high density and durability, it is suitable for industrial use. Meaning you can use this to build bridges, infrastructures, and anything you think that can be built. And the point here is that this mangkono tree is actually endangered already. So that is because, first of all, it is rare. Second, it has poor regenerative capacity. And lastly, it is heavily exploited. So what now if it is endangered? Of course, if it is endangered, we need to preserve this for the future generations. And how would we do that? Right? So we take a special trait or a desired trait of this organism and transfer this into another organism. And for further discussion of the special traits, let me call on the next reporter, Mr. Emanuel Sanidad.
Thank you, Ms. Rafael. So now we ask ourselves, what leads Montgomery to be highly exploited? What trait does the exploiters uh, actually want to acquire in Montgomery? So that leads us to the two special traits of Montgomery, which is having immense hardness and durability. So now you will ask us what and where specific DNA sequence that these two traits can be located in the genetic sequence of Montgomery tree. Our only answer is that we have nothing to offer. This is mainly due to the reason that even though Mangkoncho is truly endemic to our country alone and now tagged as endangered species, there is little to none study that has been actually conducted regarding Mangkoncho, especially regarding its genetics. What information we have so far regarding Mangkono tree is that it draws metal minerals out of the soil, thus having its hardness and durability, and giving its nickname ironwood. But we'll ask us now, does it mean that what we are proposing or our thought experiment is not feasible at all? Our answer is no. Way back in 2015, Futuragene, an international biotechnology company, successfully genetically modified eucalyptus. And if you will know, eucalyptus has, has or is under the same family tree with Mangkono tree, which is Myrtaceae or the myrtle family, thus giving us the idea that there's a high chance or high possibility that the genetic sequence they altered must be the same or closely near to the DNA sequence that we are looking for in the Mangkono tree. Tutora genes genetically modified eucalyptus produces 20% more wood compared with its conventional variety, and it is ready to harvest in 5.5 years instead of 7 years. What they did is that they inserted into eucalyptus a gene encoding called Arabidopsis thaliana, protein that facilitates cell wall expansion and accelerates growth. This was approved for commercial release by the Brazilian National Technical Commission on Biosafety in April 2015, as well as in some states of United States of America, as well as in South Africa. The successful modification, the same species in the same family tree can give us lead to where specifically this hardness and durability trait we are looking for in Mangkono can be located in its DNA sequence. So now, may I call on Ms. Riguer to discuss the target organism of our thought experiment.
Moving on to our target organism. So for this, we chose Cocos nucifera or the coconut tree. So some of us may be aware that the coconut tree is also known as the tree of life. And it is known for that because every part of the tree can be used for resources, for anything to help sustain a human's life. And coconut trees are actually abundant in tropical countries like the Philippines as it is the best and most favorable weather conditions for them to grow properly. The tree is also considered to be very versatile as it can provide its fruits for more than 60 years of its lifetime. And this is due to the fact that these trees mature very slowly. And like I said, the coconut tree is very useful as its parts can be used into different things. So the coconut tree actually produces something called coco lumber. And coco lumber is abundant, um, one of the most abundant and most used sources of lumber in the country. And we specifically chose this organism as our target because we are aiming to increase its durability, which would probably lead to the increase of efficiency and demand for coconut trees. And with this, there will be a lot of benefits to this as it could lead to stronger furnitures, stronger infrastructures, and it could also lead to the development of the country's economy due to the high demands of coconut trees or cocoa lumber.
There are various ways of transferring the desired trait of the macono tree to the coconut tree. The first would be the gene gun, wherein this gun fires plastic bullets that have tips of micro pellets that are made of gold and tungsten and are covered with the desired trait. And this bullet is fired fast enough so that it will penetrate the cells, and then DNA fragments will be left that will be later on be incorporated through the cell's nucleus. The other process would be the Agrobacterium tumefaciens, wherein there's a biological vector, an agrobacterium, that is soil dwelling. And then the nature takes its place, seeping the DNA would be seeping into the plant, and this process is taken advantage of genetic engineers, wherein they just cancel out the disease caused. Transgene insertion is generally an efficient method, as only a few or a percentage of these cells actually assimilate the foreign gene. Now, this is sufficed through making cultured tissues or cells, wherein these cells are grown at a lab and they are satisfied with specific conditions. Now, that is for the coconut tree, where it is the tree of life but better. It is still the coconut tree that still gives us everything it has, but it has sturdier lumber, which is much more useful. So that is all. Thank you for listening.